So it's my pleasure to introduce the next speaker, Dr. Beatrice Sim. Keeping to the theme of 2014, I've known Beatrice since she was our medical student in 2014, where I was an ID trainee. But more importantly, she came back to the department in 2020 and was important to my own sense of well-being in my first year as a consultant. After that, she went on to some fabulous things. She did an immunocompromised host fellowship in Melbourne initially, and then a transplant ID fellowship at Duke, where she worked with Dr. Wolf, Professor Wolf. And most importantly, we have the privilege of calling her our colleague now. She's come back to Brisbane to work with us. She'll be talking to us today about CMV transcriptomics.
Thanks very much, Lana, and thank you for the introduction and for your tolerance of what sounds like a particularly tenacious stalker. So I think we started the day off with talks about viral infections, and the plan is now to return back to them. And specifically, I'm talking today about CMV and the host response and how we can use the host response to assess patients clinically and their particular risk of CMV infection. And we'll be doing that, well, I'll be doing that today through some both published and some preliminary results as part of my PhD studies. So let's start with some universal truths. CMV, particularly focusing in allotransplant, both bad and common. So in the literature, the incidence is up to perhaps 37%. And I'm sure to this audience, it comes as no surprise that it has a range of clinical syndromes associated with it, all the way from asymptomatic viremia to clinically significant CMV infection, which the international consensus guidelines defines a CMV requiring treatment, all the way to an organ disease, which fortunately I think we see a bit less of now. And not only those direct complications, CMV is also associated with a bunch of different indirect viral complications, including things like graft dysfunction, rejection, GVHD, as well as increased non-relapse mortality. So CMV, bad and common. We've done plenty of work over the past years to try to work out how to risk mitigate this virus. And in general, we base that on serological viral dependent assessment. There are two sort of main strategies when it comes to combating CMV. One is the preemptive strategy, which has traditionally sort of been more common in the allotransplant population, though certainly we're moving a bit away from that now. And the other is the prophylactic strategy, which once again, typically more common in solid organ transplant population, although there's increasing literature that you can in fact use preemptive monitoring in the liver transplant population, and that's both safe and well tolerated. There are issues with both, as there are with many things. Some of the issues with preemptive treatment is this unclear treatment threshold. There's no sort of consensus as to, I think previously we were looking at maybe 1,000 international units per mil, and that's when we'd consider treatment, but that's sort of gone out of vogue now. How long do you treat for? I mean, there are some recommendations around 2 undetectable viral loads, but also the threshold for detection is so variable across different labs that I don't think this is particularly all that helpful or universal. And ultimately, the real issue is that we can't identify patients who are at future risk of reactivation after you stop treatment or even before you start treatment. In terms of prophylaxis, it's certainly in the allotransplant population, we use a lot more letermovir for prophylaxis now, up to 100 days, based on a NEGM trial that showed better outcomes, less clinically significant CMV infection up to week 24 post-transplant. But pushing out the risk of reactivation potentially means that you're just going to get it later. So that risk of late reactivation after you stop prophylaxis may be an issue or could be an issue. And then the other thing as well is that not all patients are going to have the same risk of CMV infection, and certainly you run the risk of overtreatment when you prophylax everyone. Even in that index NEGM paper, in the placebo arm, over 50% of the patients who were in the placebo arm, so didn't get any letermovir, didn't actually get clinically significant CMV reactivation. So clearly not all patients are equal. I would also be remiss if I didn't mention that the overarching issue is with any drug, side effects of medication and antiviral resistance as well. So who is at risk? The answer is complicated. It's especially in the allotransplant population where potentially the immunosuppression regimens are not linear or potentially not as linear as the solid organ transplant course. We have to consider both the host response to CMV as well as what we're doing to the patient. So in terms of the various arms of CMV defenses, we know that there's probably some element of humoral immunity involved, though potentially not as much as, what's traditionally the cellular defense arm. We know that we can give or have given CMV hyperimmunoglobulin in patients to prevent in pregnant patients who have primary infection, occasionally in solid organ transplant recipients as well, and sometimes in neonatal transmission, maternal transmission too. In the allotransplant population, on the other hand, I don't think this is as clear-cut. And when I say as clear-cut, it's not really clear-cut at all. Certainly, there was a meta-analysis that looked at immunoglobulin prophylaxis administration in allotransplant patients. And while it showed that there was less CMV disease, the rates of clinically significant CMV infection were approximately the same. Cellular defense, I think no one would argue with me when I say that that's particularly important in CMV infection. We know that patients with HIV, with AIDS, are all at risk. We know that patients who have stem cell transplants are at risk, lymphodepletion and lymphoid depleting therapies are at risk. And there's certainly a role of both the CD4 and CD8 T cells in managing this. And I think approximately 10% of CD8 T cells actually are CMV-specific T cells, which is quite a large proportion when you consider the range of pathogens that these cells have to respond to. And in terms of the cytokine milieus, it's well documented that type 1 interferons are very important in management of CMV, and certainly there's a importance in the IL-6 and axis as well. And all of that has to be balanced with the fact that we're immunosuppressing these patients. And because of that, they potentially would have delayed immunoreconstitution, so different timings of their risk. They might get graft versus host disease. We might have to immunosuppress them more for it. So I posit that there is an unmet need for some clinically meaningful biomarkers to predict those who are at risk of complications, both to prioritize early intervention in those who are at risk and to decrease potentially harmful antiviral exposure in those who are not at risk. And ultimately, pie in the sky, what would be ideal is that if we were able to individualize and tailor these management strategies per patient. But I think we're still a bit of a way off. So some of these strategies that we've used so far have included cell-mediated immunity assays, and by which I mean primarily these three. So intracellular cytokine staining and tetramer multimer staining is primarily used in the laboratory. So ICS is looking at the CMV, in this particular case, derived or directed cytokines, so somewhat of a more quantitative or functional assay. And the tetramer multimer staining is looking at the numbers of CMV-directed cells. Once again, those two are probably more laboratory-based, primarily because they're quite, as the name suggests, laborious. But there are some tests coming out. I think Viracor has one that combines those two assays, but the studies for them are pretty small at this stage and just confined to abstracts. What is probably more relevant to us, especially in this precinct, are the cytokine production assays. These are classically interferon gamma release assays, which is something we're familiar with in the form of the quantiferon TB test. And Rajiv Khanna, who works out of this precinct, I think was instrumental in coming up with the quantiferon CMV, which is what we have here. Overseas, they have the ELISpot assay, and particularly Rochamale in Texas has been quite successful with using that. So a quick refresher for anyone who doesn't know, the way that the quantiferon CMV test works is very similar to the quantiferon TB tests, pictured up in the left. I don't know, I've got agnosia there. It's a picture of the three test tubes. So what happens is, firstly, you've got a mitogen, which becomes important later, and that's a positive control. So it's to assess that your cells can at all produce interferon gamma to a mitogen, something that can stimulate an immune response. If you can't, the whole test is null and void. It's called indeterminate. We go no further. Then the next bit is looking at the nil tube, so looking at the patient's baseline production of interferon gamma, and you subtract that from the test tube to come up with a number. And if the test tube contains peptides from, in this case, CMV, quantiferon gallets, TB. And if you reach a threshold, then it becomes, you get classed as positive. So you have an immune response to this pathogen. If it doesn't, it's a negative test. All right, so what we've done so far is to look at the usefulness or the clinical utility of the quantiferon CMV test in the allotransplant population. And that was done at 4 sites in Australia across about 2-3 years. We included all adult patients with CMV recipient seropositivity undergoing allotransplantation, and excluded those who were lower risk or those who had active disease, so CMV disease at the time. What we essentially did was we monitored these patients from baseline and collected blood samples, including quantiferon CMV, at approximately 6-week intervals after their transplant. And if patients had clinically significant CMV infection, by which I mean, once again, that requiring treatment, we collected an extra sample about 2 weeks into their treatment course. So this was the population that we used. So there were 119 patients in total across a variety of different primary diseases. And of those patients, 54 of them had clinically significant CMV infection. And that was 1 episode in 44 and 2 episodes in 10. It was approximately a median 51 days at median to first clinically significant CMV infection with a peak viral load over 4,000. CMV disease, which is consistent in our population, was relatively uncommon. There were only 6 cases and they were all gastrointestinal. So across the study period, we performed 385 tests, a median of 3 tests per patient. And what I think is notable is that not many people had a positive quantiferon CMV result. So only 40% at any point post-transplant had any positive CMV result, quantiferon CMV result. And of these patients, only about 16% had multiple positive results. And what potentially is more notable is that about 14% of our population had what we call stable reactivity. And what I mean by that is that if you to have stable reactivity, you had to have 2 consecutive positive quantiferon CMV results and maintain that, so you wouldn't have a negative result thereafter. So are you able to mount a response to CMV, and are you able to maintain that response? And honestly, it was pretty dire. So you can see in the alluvial plot on the side, the reactive tests are in yellow, and they're quite vanishingly rare. Certainly there's lots of crossover between states, and I think that's reflective of the fact that the net immunosuppression state of the patients varied so much. So what I think we can get from that is that quantiferon CMV is quite a dynamic test. It's not one that we can use at a predetermined time point. But what it does predict is peak viral load per test, and this was consistent over each of the time points. So we did a mixed effects regression model, classic for patient, and what we found that is if you had a positive quantiferon CMV test, then you would have, it was associated with at least about a 0.5 log lower peak viral load in that sort of 6 weeks post that test, and then it reset again when you had another test. But what we really care about is clinically significant CMV infection. Like, do we actually care about this viremia in this patient? And in general, what we found was that the quantiferon CMV result was not associated with clinically significant CMV infection, and certainly not the baseline, which has been shown in some of the solid organ papers, but also not in the post-transplant time period. What we did find, though, which was quite interesting, was that if you had a higher mitogen tube production of interferon gamma, that was significantly negatively associated with clinically significant CMV infection with a hazard ratio of 0.92, by which I mean, if you with every 1 international unit per mil increase in interferon gamma response to the mitogen, you had a corresponding about 8% decrease in risk of clinically significant CMV infection. And that was consistent when we considered all the biologically plausible variables. But is that all that we can learn from this particular cohort of patients? Hopefully not. So we pulled out the clinically significant CMV infection patients and did RNA sequencing on their samples to look at real-world transcriptomic profiles over the course of infection, and also maybe, hopefully, to identify the differences associated with what we would term a more complicated infection. So this is what we did here. So we had we took 2 samples per patient. We did one at baseline, so prior to CMV reactivation, and that 2-week into treatment time point. We split the cells and we stimulated half of them with a mitogen to look at their responsiveness overall. And we used PMA and ionomycin for that. And we left the other half alone, unstimulated. And we did RNA sequencing on both sets. So changes over treatment. So as expected, there were a whole bunch of genes that were differentially expressed between your post-treatment time point and your baseline time point, about 4,000 genes, and the top 10 are sort of represented in that table there. In general, upregulated genes were markers of adaptive immunity and protein transport. And when we did hallmark gene set analysis, that generally demonstrated a reduction in that pro-inflammatory pathways post-treatment. So overall inflammatory response, your NF-kappa beta pathways, IL-6 that I mentioned earlier, and of course the interferon gammas as well. But when we went to look at the multiple infections, that probably doesn't come up very well on the slide, sorry. When we went to look at the multiple infections, what we found was that with the exception of potentially the E2F pathway, which has been associated with a reduction of CMV replicated in vitro anyway, the pathways were grossly downregulated, or the inflammatory pathways were grossly downregulated post-treatment as compared to baseline. And despite administering mitogen, so ionomycin and PMA, this did not restore the balance of inflammatory pathways. So potentially reflective of the fact that these cells were not responsive in general. So what about peak viral load? So what we found in this particular case was that unstimulated samples with higher peak viral loads demonstrated upregulation of those type 1 interferon pathways, so interferon alpha, but downregulation of all the other inflammatory pathways. But in this particular case, it was completely reversed when we stimulated the samples, so potentially reflective of a more dysregulated hyperactive immune response. So to conclude, CMV is a difficult infection to predict, risk stratify, and treat. And while there are new clinically available assays that provide some nuanced information on cell-specific immune reconstitution, I think there's still much that we can learn. There are differences in the immune response between patients who develop recurrent CMV infection or worse disease, which I think it's probably early days yet, very preliminary data, but potentially there is some potential for development of an immune signature to predict CMV severity. So watch this space. Thank you to all my collaborators and my supervisors, including Cam, who's in the room.